Before I get into that, I do want to talk about a couple of things. So first off, your chapter 2 outline, you should have turned in by now. If you haven't, definitely turn it in. And if you haven't turned in your rankings for the disease project, please turn those in by midnight tonight. I'll try to get those groups put together. Yes. It should be in the assignment. So if you click on the assignment, there's a description of it. And then at the bottom, there's a list of diseases. Yeah, I probably should have put that in the spreadsheet. That would have been helpful. So yeah, get that turned in and hopefully I'll get everybody grouped up by, hopefully you'll know by Wednesday. And then also I'm going to offer extra credit for attendance at either SNAP groups or one-on-one tutoring. So how I'm going to do that. So the SNAP groups are starting up this week, I think. I'll put the schedule on Zoom, on Canvas. And if you go to 2 meetings, it can be 2 SNAP groups, 2 one-on-one tutorings, 1 of each, it doesn't matter, but you have to go to 2 before the first exam, which is next Friday. You will get 2 bonus points on your exam, and that's out of 50 points. So it's not out of 100 points, so it's 2 points out of 50. So I'll get the attendance from, you know, whoever's in charge. And I think those, I want people to go to more than 1 because I think it's helpful if you do repeat visits. And I've seen people who have gone kind of multiple times really improve their test scores. So if you think that's helpful to you and you want those bonus points, definitely do that before the first exam. I'll kind of reevaluate after the first exam and see if I'll offer them again, but I may not. So this might be the only chance you have. So like I said, I'll put the SNAP group schedule on Canvas, and then the one-on-one tutoring is just something you set up on your own. Any questions about that? All right, let's jump back into chemistry here. All right, so we ended with water. I don't think we quite got all the way through this video, so definitely watch it and focus on these 4 properties of water that are listed here as particularly important, at least from an AMP perspective. There's a lot of properties of water that are important, but definitely from a human health standpoint, these 4 properties are important and be able to explain them and kind of pick out some examples of them. One more slide on some basics of water. I think I've said this already, and most of you probably already know this, but this is a characteristic you definitely want to know. Water is polar. It's polar, which means there's a slight negative charge around the oxygen and slight positive charges around the hydrogen. That means it's going to be attracted to other polar substances. So likes mix with likes is an easy way to think about it. Polar substances will mix with polar substances. Water being polar is not going to mix or bond with anything that's non-polar. So if there's no charge, no partial charges on a molecule, like in lipids, oils and fats, then it's not going to be attracted to it and it's not going to bond with it. So you know oil and water doesn't mix. That's the best example of polar and non-polar substances not combining. So this is really important in our body, obviously. So I wanted to separate that out in particular because water being polar gives it a lot of its specific characteristics that I listed on the previous slide. Okay. Would you mind closing that door? Thank you. If I can hear it, I'm sure you guys in the back are really distracted by it. Thanks. Okay, so let's get into how water mixes with some other stuff. So mixtures in general are going to be, we're going to talk about a few different types of mixtures. And I know this sounds sort of not very human oriented, but our body is just a mixture of a bunch of different stuff, mostly with water as the base. So we're going to talk about 3 different types of mixtures that occur in our body. First, what is a mixture? It's just something that's physically blended, but not necessarily chemically combined. So this includes most of our body fluids, Saliva, urine, and then, yeah, our blood is a specific kind of mixture. So chemicals dissolved or suspended in water. That's what we're talking about here. Water is always going to sort of be the base, especially from a human standpoint. An interesting note, guys may have heard this before, but up to like 3/4 of your body weight is actually water. So it depends on your gender, your age, your weight, but it could be as low as 50% or up to 75%. So somewhere between 1/2 and 3/4 of our body weight is actually water. So that's what's kind of holding on to all of these other dissolved and mixed in substances. Now I'm going to go into 3 specific kinds of mixtures. So solutions, colloids, and suspensions. The way you distinguish these technically is based on the solute. So if you guys remember, a solution is made up of a solvent and a solute. The solvent does the dissolving, so we're going to think of that as the water, basically. And then the solute is what's getting dissolved. So the size of the solute, if it's really tiny, under 1 nanometer in size, so under 1 nanometer in size is going to be, that's going to make it a solution. That essentially means that light is not scattered. You can see all the way through it. So you can see all the way through the solution, and the particles are evenly distributed throughout, so it's homogeneous. You don't see little chunks of dissolved or partially dissolved particles. It just looks all the same. So salt water is a good example of that. If you dissolve a bunch of salt in water, you dissolve it enough to where it's actually dissolved, that's a solution. It doesn't separate on standing either. So that's going to distinguish solutions from suspensions in particular. So if you leave salt water out, it's not like you get a bunch of salt at the bottom and then water on the top in most circumstances. So that's a solution, kind of I guess the most obvious sort of mixture that you would think of. Good. And then colloids. So colloids are that kind of in between solute particle size. So solutions had solutes of under 1 nanometer. Colloids have solute particles between 1 and 100 nanometers. And I know you probably don't have a good sense of what a nanometer is. It's tiny. So just kind of, that's sort of the technical definition. That size is the technical definition. Identifying them, that's why I gave you these other characteristics. Are they homogeneous? Do they scatter light? That kind of stuff. But you want to know all these bullet points, basically. So the technical definition, that size, as well as some of the physical characteristics, where if you looked at a test tube of something, you would know if it's a colloid or a suspension or a solution. So colloids are the in-between. They are going to scatter light, which just means you can't see through them perfectly. That's what I mean by scattering light. And I'll show you an example of these a couple slides away. And they're usually cloudy. So milk is a great example of a colloid. So larger particle sizes basically don't let light completely penetrate it. And still, just like solutions, the particles remain permanently mixed. They don't settle out. If you leave them over time, milk, ideally, unless it's disgusting in some way, there's no solid particles that settle out, right? It stays in the mixture. Milk, I think, is the best example in our body. Colloids usually consist of protein plus water. So different proteins plus water. Proteins are kind of medium size, definitely larger than, say, sodium and chloride ions, but they're not gigantic to where they'll actually like settle out on their own. So protein and water is a good example of a colloid in our body, even though it's kind of hard to relate to. All right, so we got solutions, colloids, and the last one is suspension. As you might guess, this is the category where particles are actually really big. Anything over 100 nanometers in size is technically, if the solute is over that size, it's considered a suspension. They can appear cloudy as well, or just totally opaque. And opaque just means you can't see through them at all. The really important part here, and it's kind of easy to understand from the name of it, is that the solutes, the large solutes within the mixture will settle out if you just let it sit there for a long time. So if you don't keep mixing it, the solute particles will settle out to the bottom and there'll be some separation within the suspension. That separation is called sedimentation. So you get a sediment at the bottom, say, of a test tube. The best example of this is our blood. So our blood is a mixture of, well, a number of different things, but in particular blood cells. So we have red and white blood cells. And then plasma is the liquid that they're suspended in. So you have plasma with a bunch of red blood cells. We'll just imagine the red blood cells scattered throughout. That's a suspension. Blood cells are pretty big. So they're going to settle out over time. If you leave a test tube of blood, over time, you'll see the separation between the blood cells and the plasma. Okay, so let's look at some examples here. So I covered up the answers, but the diagram, the drawings on the right are just kind of representations of what's going on the left. So same thing. This is represented by this in the drawing. So you guys on Zoom, this blue is the same as this test tube drawing here. So which of these, we'll start with the easiest one. Which of these would you say in these final two are the same substance, but this is over time? So time 0 and then time, we'll say 12 hours in advance, like further down the road. So which of these would be considered a suspension? D, yeah. So C and D are the suspension. So this is blood, basically a test tube of blood. And then over time, so at first when you draw blood, it looks just like normal blood. If you leave it there for a few hours, it'll settle out into the plasma and then the blood cells on the bottom. So that's a suspension. That's how you recognize a suspension. And you can see it's totally opaque. You can't see through it at all. Okay, and then what would be we'll go to the other side, solution. Which one would you say is the solution? A, yep, A is the solution. You can see through it. So you can see the lines behind the test tube. So totally see through, that's going to be a solution. So for you guys on Zoom, this is our solution, the blue. They just added some food coloring to it. And then this is going to be our colloid. So you can't really see through it, but it hasn't settled out. You just have to take my word for that. It doesn't settle out over time. So that whitish color, the milky solution, the milky mixture there is the colloid. So if you, I just blacked out the answers over here. So on the PowerPoint online, you can move that black box and see the actual answers. So that's how you sort of visually distinguish the solutions, colloids, and suspensions. All right. So this we're actually going to talk about a good bit in lab this week, so I'm not going to spend too much time on it. Acids, bases, and pHs, I think everyone has a general idea of what pH is, right? So the important points that I want you to know about pH, first of all, our body, our blood is at a very specific pH within like a 1/10 of a point. It has to stay within. So if it goes off at all, it can totally throw our body out of whack. So pH is critically important, not just so we can say, yes, lemon juice is acidic and bleach is basic. Within our body, our pH has to stay very, very stable. So we're going to practice some of this in lab today or this week. But some of the main points that I want to talk about before that are that the definition of an acid is that it releases hydrogen ions in water. So H+, it's a hydrogen ion. A base on the other side is something that is going to accept the hydrogen ion. So if you think of you imagine a chemical reaction, these hydrogen ions are coming off of the acid and going to the base, basically. You can also think of a base as donating hydroxide ions, OH-. We haven't talked about hydroxide yet, but I'll get to it in just a few slides. So usually a base, if you see a molecular formula like NaOH, Sodium hydroxide, if it has OH on it, that usually means it's a base. Hydrochloric acid, HCl, it can donate that H, which makes it an acid. And then measuring pH, it's a little bit weird because it's a negative log scale. We don't deal with log scales in most of our lives. We kind of deal with linear scales. A log scale is a little bit weird. And the fact that it's a negative log scale makes it even weirder. So pH basically stands for percentage of hydrogen ions. That's why the H is capitalized. So how many hydrogen ions there are in solution. That's what you're measuring when you're measuring the pH. However, the more hydrogen ions there are, the lower the pH is going to be. So it's flipped. So more hydrogen ions means it's acidic. If we come down here, so pH, so 7 is sort of our cutoff. A pH of 7 is neutral. That means the hydrogen and hydroxide ions are balanced. There's the same amount. If the pH is less than 7, it's acidic. So that means there's more hydrogen ions than hydroxide. And then the opposite is true for basic. So you would expect, if we're measuring the percentage of hydrogen ions, you would think, well, if the pH is higher, that means there's more hydrogen ions. That's where this weird negative log scale comes into play. So actually, as you increase the hydrogen ion concentration, that means it's a lower pH. That's the negative aspect. The log aspect has to do with this next bullet point I have down here. A log scale sort of puts the scale at in a simpler way to understand. So if we have a pH of 1 versus 2 versus 3, it's nice and clear and it seems linear and clear cut. But if you move from a pH of 5 to 4, so we're moving in the acidic direction. A pH of 4 is actually 10 times as acidic as a pH of 5. So for every point moved on the scale, it's a 10-fold difference in acidity or basicness if you go the other way. So those are really important to understand. Lower pH actually means there is more hydrogen ions. And then 1 point movement on the pH scale means a 10-fold change in the pH, whichever direction. Does that make sense? Those are two really important aspects of pH to really understand it. Does anyone on Zoom have any questions? I don't want to forget about you guys. Okay, we'll practice some of that and do some examples in lab really briefly this week. And if we look at the pH scale, I think everyone's probably seen this, but this just kind of gives a little bit of reference with common materials that you guys are familiar with. So neutral is 7, water is neutral, pure water. A lot of water has maybe some extra base or might be slightly acidic, depending on where you're getting it from. But pure water should be a 7.0. If we go all the way down, we have, lemon juice, vinegar, all of that's pretty acidic. Our stomach acid is really, really acidic, hence stomach acid. If we go all the way over on the other side, there's not a whole lot of human aspects that are really basic, like on the far basic side, but you have a lot of cleaning products like bleach, ammonia, all that kind of stuff is really basic. Okay, so that's just a reference for you. Remember, lower pH means more acidic. Okay, and it goes from 1 to 14. So now we're going to sort of switch gears and move into chemical reactions. So we started out this chapter with some really basic chemistry, and I was talking about how atoms bond with each other. And the whole point of that is because we have these atoms bonding with each other to make these really large molecules that we know and love and survive off of: carbohydrates, lipids, proteins, and nucleic acids. So that's where we're moving with all of this information. So in general, I'm going to talk about a few different classes of chemical reactions in a very general sense. So talk about these, and then the definition of a chemical reaction is simply where an ionic or covalent bond is either made or broken. So it can be either one. So we're doing something with bonds. That's why we talked all that that's why I gave you all that information about bonds, not just for fun. I should take a minute here to say so I cut some information from this PowerPoint. So if you have like the old version, if you printed it out, I uploaded the new version last night or yesterday afternoon. Because we're running a little behind, I just kind of cut some information that I thought wasn't as pertinent to A&P. Is it important? Yes, but I thought moving forward, in order to kind of stay on track, I got rid of a few slides. So if you see some that you're following kind of the old version, if there's a slide that I don't have up here, you don't need to know it, basically. So the new version is on Canvas now. Okay, so the classes of chemical reactions, I'm really going to focus on these two, decomposition and synthesis. Decomposition is obviously breaking a molecule down. Synthesis is building a molecule from smaller pieces. So making and breaking. Exchange, that's something I sort of cut out. I want you to know that there is such a thing as an exchange reaction where essentially two kind of complex molecules will switch their partners. They'll exchange whatever they're bonded with each other. That is, if you know, redox reactions, oxidation reduction reactions, that's an exchange reaction. That's something I cut out. They're important, but I thought big picture, not critical that we cover that in this class. If it comes up further down the road, I'll explain it, but I cut out the redox stuff, which means we won't really talk all that much about exchange reactions. All right. Now just some simple diagrams of what the heck these look like in very simple terms. When we break down a starch molecule, so say we eat a potato, and we break down all that starch that's in the potato, we can break it down into individual glucose molecules, because all starch is a bunch of glucose molecules strung together. That's going to be decomposition. We're decomposing a complex molecule into its simpler parts. Synthesis will be the opposite. So to make a protein, you need amino acids. A bunch of amino acids strung together essentially makes a protein. That's going to be synthesis. And then exchange, like I said, I'm not really talking about, just means the two molecules will switch whatever they're bonded with. So they're a new combination, basically, of atoms. So decomposition and synthesis are the really important ones as we get into these biological macromolecules. Okay. So I'm gonna actually step back a little bit and do some other sort of basic chemistry, but some stuff I brushed over early on when we were talking about atoms. So I said, we don't care about protons and neutrons, right? But I do want to point out that all of these reactions are essentially determined by how many protons there are. So I said, wait, the electrons are what matters, right? Why do the protons matter? Basically, the protons in the nucleus are what's attracting the electrons. So the protons are positively charged, the electrons are negative, which means without the protons, you don't have the negative charge being attracted to the atom. So the electrons wouldn't be there at all. So what I mean by ultimately determined by protons is that you can trace it all the way back to how many protons there are. That's going to determine how many electrons there are. So because protons attract the electrons, and the electrons are what leads to the bonding, finally, really it comes back to protons. So just kind of a slight alteration to what I said, I guess, a little more explanation to how these atoms work. And that brings us to the idea of isotopes as well. So I had a lot more information on isotopes, but I'm breaking it down into the basics of isotopes for what you need for A&P, knowing that this is not a chemistry class. So what's an isotope, first of all? Basically, isotopes of an element are the same element, so say carbon, but they have different numbers of neutrons. So we also didn't really talk about neutrons all that much. This is where neutrons come in. If carbon has different numbers of neutrons, it's still carbon. It just reacts slightly differently and has some different characteristics. So there's multiple different isotopes of carbon.
That just means they have different numbers of neutrons, but the electrons and protons are the exact same. So here's hydrogen. We can ignore hydrogen, but there's different isotopes of hydrogen as well. But carbon, all the way over here, see if you guys can see. Carbon on the bottom, there's at least 3. There's multiple, there's beyond this, but carbon-12 is the normal one. So if you go over to the periodic table, there are that red number, that's the atomic number. That means there's 6 protons. So for you guys on Zoom, sorry, I don't have a periodic table, but there's 6 protons typically with carbon. That's why we see the 6 at the bottom there. The number on top is the number of neutrons plus protons. So you can see normally carbon-12, we have 6 protons, 6 neutrons, 6 electrons. Usually it's all equal. When we get into the different isotopes, say carbon-14, carbon-14 still has 6 protons, but now it has 8 neutrons. So slightly different number of neutrons, but it actually makes it radioactive. So it totally changes it. Just with 2 neutrons added, it makes carbon-14 radioactive, whereas carbon-12 is not. Carbon-12 is what's mostly in our body. Okay, so that's going to create different atomic masses, which I've noted here. So in an atom, you have protons, neutrons, electrons. Electrons basically weigh nothing. They're super light. Protons and neutrons make up most of the weight of an atom. If you have more neutrons, that makes it heavier. And that's essentially, in a very simplified way, why carbon-14 is radioactive. Basically stays around a lot longer because it's heavier. Okay. So like I said, that's sort of a step back to some of the atomic, like basic atomic chemistry, but I think you need to understand some of that to at least move forward. So any questions about that? Isotopes, basically just different forms of atoms or molecules once we get into them. Okay. Now into something that very, very directly relates to the human body, metabolism. This is where I actually cut some information out as well. So I broke down metabolism to sort of a simpler explanation. So you probably heard, oh, you have a fast metabolism or you have a slow metabolism, or we've heard metabolism mentioned in our everyday lives. From a scientific standpoint, metabolism is basically just all of the reactions happening in your body. That includes reactions that are building large molecules from small pieces, which is anabolism, as well as reactions that are breaking down really large molecules into their smaller components, which is catabolism. So over here, you can see really simple diagrams. Catabolism, you're breaking something down. That's actually going to release energy. So if you break down a big molecule into smaller pieces, you're releasing energy. If you have to build a big molecule from the small pieces, that takes energy. So it's going to use energy and thus store energy, basically, in all of those bonds. If you think about it as like building a house or a building that up takes a lot of energy, just like building a large molecule from smaller pieces. That brings us to the idea of polymers and monomers. So monomers are the individual subunits. They get bonded together, that's going to make a polymer. And we'll talk about the monomers and the polymers of these different biomolecules for the rest of this chapter. So mono means one. So one piece, poly means many. So many monomers strung together makes a polymer. And sort of an, I guess an easy-ish way, maybe a helpful way to think of, remember anabolism versus catabolism, because it's hard to remember which one's building and which one's breaking down,
if you know of anabolic steroids, they build muscle, right? If people are taking anabolic steroids, it makes them get bigger muscles, they're building muscle. So anabolism, it's the same term, basically. I don't have a good analogy for catabolism. Can't think of how that would relate, but if that helps you remember anabolism, then you know catabolism is the opposite, it's breaking down. Okay, and that brings us into the organic compounds. So some of this, at least if you took General Bio 1101 here, this will look familiar, and it won't actually even be in as much detail as we went through in General Bio for these different carbon compounds. If you haven't had General Bio or took it somewhere else and maybe didn't go over these, this might be kind of new, but hopefully for most people, it's sort of a review. So organic chemistry, has anyone taken organic chemistry in here? Okay, I was terrible at organic chemistry. I did not understand organic chemistry. Every other chemistry I was fine at, but organic chemistry is like another world. Thankfully, you don't really need to know all of the details of organic chemistry in order to understand these 4 organic compounds. I'm going to tell you what you need to know from a biological, human anatomy and physiology standpoint. So that's good news. Organic chemistry in general is just a study of compounds with carbon. So something that's organic typically has carbon in it, usually hydrogen as well. But carbon is the main part of organic chemistry. So we're going to talk about 4 different categories. Most of these I assume you've probably at least heard of. So carbohydrates, lipids, proteins, and nucleic acids. We're going to talk about what they look like, kind of how some of their characteristics, and why they're important in our bodies. So we'll go through those one by one. Before we get into that, I think I already mostly defined these, but I wanted to have these definitions up here for you to reference. When we're talking about macromolecules, these are all macromolecules. So we're not talking about a tiny water molecule here. We're talking about really big molecules in our body, well, relatively. For a molecule, these are going to be big. So macro means big, so large molecules. And then monomers and polymers, I'm going to talk about the monomers, if there are any, for each of these categories of molecules. So those are the individual subunits. And then the polymers are just a bunch of those subunits strung together. That's the final molecule that we're going to get to. So proteins, like I said before, are made up of just a bunch of amino acids strung together. Okay, so let's get into, oh no, just kidding. Let's get into functional groups, not carbohydrates yet. So this is another sort of some information that you need to understand these macromolecules in addition to their bonding and the monomer polymer thing. So there's a lot of functional groups in chemistry, which are basically just groups of atoms that you often see together. So they're often bonded to molecules in a similar fashion. So you'll see hydroxyl groups coming off of these molecules, which are just O and H. So OH is hydroxyl. So what you need to know from this chart, the name of the functional group, and be able to identify the structural formula, which is just essentially the atoms that are involved. So hydroxyl is OH. Carboxyl is COOH. It doesn't really matter what order you write them in. You don't really need to know about the importance. That's just sort of for your information if that helps explain them a little bit. So hydroxyl and carboxyl. Amino is easy because it's the only one with nitrogen in it that you need to know. So N plus 2H is bonded to that is going to be an amino group. Methyl is where there's a C with 3 hydrogens, carbon with 3 hydrogens coming off of it. Those are really, really common. So if you know methane, it's CH4, so methyl. is CH3. And then phosphate. Phosphate comes into play in a large way when we're talking about cell membranes as well as ATP. If you remember, ATP is the main like energy source for our cells. And that's a phosphorus with 4 oxygens coming off, and it has a -2 charge. You don't really have to worry about the negative charge. But phosphate with 4 oxygens, that's phosphorus with 4 oxygens is phosphate. Those are the only 5 I really want you to know in here. There's a lot more, but those are the 5 really important ones when it comes to human physiology. All right, now let's get into carbohydrates. Everyone has heard of a carbohydrate, right? You've heard the word carbohydrate. Are they good to eat? Are they bad to eat? Who knows? I'm sure some people know. I'm sure in moderation, like everything, they're good. We need carbohydrates for our body. So a lot of our body functions on carbohydrates. If we break it down to sort of the molecular level, all a carbohydrate is a molecule that has some version of this formula. And all this is with CH and O. So they only have carbon, hydrogen, and oxygen. You won't see any other elements in there. What this sort of complicated looking formula means is that for every 1 carbon and oxygen, there's 2 hydrogens. So double the number of hydrogens for the number of carbon and oxygen. So in glucose's case, there's 12 hydrogens and only 6 carbons and oxygens. So you just double the hydrogen. You do want to know the molecular formula for glucose. So glucose is really important. It's what is our blood sugar. So it keeps us kind of awake and functioning correctly. C6H12O6. You want to know that that's glucose. These molecules are hydrophilic, which means they are water loving. So philic means loving, phobic means fearing. So we'll talk about hydrophobic molecules in a little bit, but these molecules mix fine with water. They don't separate out from water. And then if we're talking about the monomers and the polymers, the individual subunits that are strung together to make the overall carbohydrate are called monosaccharides. A bunch of those together are called polysaccharides. So the ultimate carbohydrate, like when you get to the final larger kind of energy storage forms of carbohydrates, those are polysaccharides. And anytime you see this OSE, which you'll see a lot with carbohydrates, that indicates that it kind of has a sweet flavor, sugar or sweet. So glucose is our blood sugar, sucrose, fructose, all of that kind of stuff. Those are all carbohydrates. All right, so that's the basics of carbohydrates. I'm going to ask you to know a few monosaccharides and then a few disaccharides. So carbohydrates are the only place where we actually talk about 2 monomers stuck together specifically, but first let's do the monosaccharides. These are the individual subunits. When they're strung together, when a bunch of them are strung together, they'll make our important biological macromolecules. The 3 important monosaccharides are glucose, galactose, and fructose. And they're actually just structural isomers. So an isomer, kind of like an isotope, is essentially the same molecule, just arranged a little bit differently. And you don't need to know how they're arranged differently. You can look at the ring structures over there if you want to. You don't need to know the ring structures of these 3 monosaccharides. I just put them over here for reference. But do know they are all, the molecular formula, if you break it down, they all have 6 carbons, 12 hydrogens, and 6 oxygens. So like I said previously, well, if I give you that molecular formula, it could be any of these 3. It could be glucose, it could be galactose, it could be fructose. And I'm not going to have you recognize them, like I said. Just know that they are all monosaccharides, They all have the molecular formula C6H12O6, and they're isomers of each other. So depending on the combination of these three, we get a couple of different, I think 3 different disaccharides that I want you to be familiar with. So di means 2, obviously. So we're going to put 2 of these monosaccharides together and make some other molecules you need to know. Those are going to be sucrose, lactose, and maltose. Sucrose is table sugar. That's going to be glucose and fructose. So you have 1 glucose monomer plus 1 fructose monomer, stick those together, and you get your sucrose, your table sugar. Lactose is the sugar in milk. You might be lactose intolerant if you can't digest that sugar well. That is glucose plus galactose. So sort of easy to remember. We have galactose making up lactose. And notice that glucose is in all of these, right? So glucose is critical to all 3 of these disaccharides. So we have sucrose, lactose, and then maltose is used in grain products like beer. So if you describe beer as malty, it's from this maltose specifically, from that sugar. And maltose is just glucose plus glucose. That's the easy one. 2 glucoses strung together makes maltose. I put the ring structures over here again so you can see each of the individual monomers. So maltose, 2 glucoses, lactose, etc. So you can see the 2 rings put together, but you don't need to recognize them by the ring structure. Just know sucrose is made up of glucose and fructose. Just know the bullet points here. Know that it's table sugar, and then lactose is the sugar in milk, maltose is the sugar in grain products. So know where they are, but you don't have to recognize those rings. Any questions about that? All right. Then the polysaccharides. There's only 3 polysaccharides that I want you to be familiar with. These are the 3 that are important from a human perspective. But weirdly, 2 of them are found in plants. But we eat plants. We eat a lot of plants, and we eat things that eat plants. So they end up being important to us as well. So first off, glycogen. You don't have to worry about the structure or kind of what monomers go together to make these polysaccharides. Just know it's a bunch of monosaccharides strung together. So more than 2, definitely. So really long chains of monosaccharides. So here you can see. So glycogen is going to be our storage form of carbohydrate. So if you eat a lot of carbohydrates and you don't need them right away, they form into glycogen and maybe eventually fat, but that's a lot further down the road. So glycogen is where carbohydrates, it's an energy storage form, carbohydrates in animals. Starch and cellulose are both in plants. So like I mentioned earlier, if you eat a potato, that's a bunch of starch. That's the plant storing a lot of its energy, a lot of its carbohydrate energy in that tuber, in the potato. So starch is an energy storage form in plants. So both of those first ones are energy storage, one in animals, one in plants. The last one, cellulose, is a structural carbohydrate, which just means it's really strong. So the energy storage forms aren't necessarily strong. They just are good at storing energy in their bonds. Cellulose is really, really strong. It's what keeps plants in a lot of ways upright and keeps their cells pretty solid. So why do we care about it? Basically, that's what we're in the dietary fiber. You're supposed to eat a lot of fiber and it's healthy for your digestive system. Dietary fiber essentially is cellulose. So cellulose is really hard to break down for most organisms when they eat it. And thus, when it doesn't get broken down, it's really good at cleaning out the digestive system, just scrapes it clean. So that's what fiber does. So you can trace it back to that cellulose polysaccharide. Any questions about those three? What you need to know or anything else about them? Okay, we're gonna end there today. I think that's pretty good. Yeah, we'll get into lipids. We'll get into the last three on Wednesday. So I will see you guys either in lab or I'll see you Wednesday.